Module 3. Now that you've learned about your red and white blood cells, let's talk a little bit about platelets. Platelets are disc-like fragments of larger cells called megakaryocytes. One megakaryocyte, on average, forms about 4,000 platelets. Now, platelets do contain some organelles. They possess mitochondria, lysosomes, cytoskeleton, and numerous granules containing clotting factors. But because they're only cell fragments, they do not possess a nucleus. Platelets are your second most abundant formed element. You have about 130,000 to 400,000 per microliter. They are your smallest formed elements. They are only about 2 to 4 microns in diameter. They live only 5 to 6 days when freely circulating in your blood. 25 to 40%, however, are stored for longer term inside your spleen. Now, if you do have a platelet count that is over 1 million per microliter, you're suffering from what is called thrombocytosis. So again, thrombocytosis is a platelet count over 1 million per microliter. This accelerated production of platelets is usually in response to a severe infection, inflammation, or cancer. If your platelet count is below 100,000 per microliter, you're looking at a disorder called thrombocytopenia. Thrombocytopenia is usually caused by red bone marrow destruction due to radiation, drugs, poisons, or leukemia. The issue that people have when they do have thrombocytopenia is that they can't clot effectively. And if you can't clot effectively, it makes bleeding that much easier and that much longer. Now, when you talk about platelet development and you talk about thrombopoiesis, as with all of the other formed elements, it's going to begin with hemopoietic stem cells. These hemopoietic stem cells divide, and one of the children becomes a megakaryoblast. The megakaryoblast duplicates its DNA repeatedly without dividing, forming an extremely large megakaryocyte. In fact, the megakaryocyte is the largest cell that can be found in the red bone marrow. As you can see in the lower figure on the screen, the megakaryocyte actually extends long tendrils called proplatelets from the red bone marrow through the capillary wall and into your blood. The proplatelets shear off and break into platelets. Now, platelets perform a variety of different functions. They secrete vasoconstrictors, meaning they secrete chemicals that can decrease the diameter of your blood vessels. And this is important, especially in reducing blood loss after injury. They form temporary platelet plugs that help to seal small breaks in broken blood vessels. They secrete procoagulants or clotting factors, which help to promote blood clotting. They initiate the formation of a clot-dissolving enzyme, which helps to dissolve clots after healing is over. They secrete chemicals that attract neutrophils and monocytes to sites of inflammation. They can even internalize and destroy certain types of bacteria. Lastly, they secrete growth factors that stimulate mitosis, or cell division, in fibroblasts and smooth muscle to help promote the repair of damaged blood vessels. Now that we've introduced what a platelet is and what on average it can do, let's turn our focus specifically to hemostasis. Hemostasis is the sequence of responses that occur in the body that help to stop bleeding. Hemostasis involves three separate mechanisms. You have vascular spasm, platelet plug formation, and coagulation. Vascular spasm is the first mechanism. It occurs immediately after damage and can last up to about 30 minutes. It involves the contraction of smooth muscle in the walls of damaged blood vessels. This contraction decreases the lumen, or the opening within the blood vessel. Now, the contraction is due to and can be triggered by physical trauma to the smooth muscle itself. Smooth muscle is very sensitive to damage, and whenever you damage smooth muscle cells, they respond by contracting. The contraction is also triggered by serotonin, a chemical released by your platelets. It can even be caused by the stimulation of pain receptors that actually can lead to vasoconstriction in nearby blood vessels as well. The purpose of vascular spasm is to make that lumen, that opening in the blood vessel, as small as possible in order to reduce blood loss. Now, in addition to the smooth muscle contracting, there are also changes occurring during vascular spasm to the endothelium of your vessel. The endothelium is the inner lining of your blood vessel, and that inner lining is made out of endothelial cells. So after injury, as the smooth muscle is contracting, the endothelial cells also contract. And when they contract, they expose a structure called the basement membrane. Now recall from A&P1 that epithelial tissues always are associated with a basement membrane, a layer of protein that helps the epithelial tissue to attach to connective tissue next door. So what's happening here, the endothelial cells in the endothelium, which is a simple squamous epithelium, contract, and they expose this basement membrane. Now, what's most important in this basement membrane is the protein collagen, and it's exposing these collagen fibers to your blood as it is passing by. Your endothelial cells also release a lot of chemical factors and hormones that actually can intensify your vascular spasm. So they're going to make that contraction even stronger, decreasing the size of your opening even more, reducing your blood loss even more. The chemical factors and hormones also have the ability to increase cell division, which is something that you want to start promoting at the very beginning, because it's the increase in cell division that's going to help you repair that damage that was induced. The endothelial cell plasma membranes at the break also become very sticky. And because we're making our lumen, our opening, so very small, the plasma membranes of the endothelial cells start to stick to each other. And when they start to stick to each other, they actually can completely seal off your break. Also, by having this stickier surface, it's going to help promote platelet attachment and begin our second phase of hemostasis, which is platelet plug formation. Now, normally, the inside of the blood vessel wall, so that lovely endothelium, is very smooth, and coated with a chemical called prostacyclin, which is a platelet repellent. However, thanks to vascular spasm and the changes in the endothelium, we have changed this smooth surface that repels platelets into a sticky, tacky one that actually allows us to do the first step of platelet plug formation, which is platelet adhesion. Platelets that are flowing by are going to start attaching to this rough, sticky surface. Now, once the platelets attach, they start to reach out with their plasma membrane and start making cytoplasmic connections with each other and with the vessel wall. Now, this interconnected mass of platelets is the beginning of what is called a platelet plug. It's a soft mass of platelets that can reduce or stop minor bleeding. However, it's a very soft mass. It's not strong, and it's very easily dislodged. So it's very easy at this point to still initiate bleeding if there's any tension applied to the wound. Now, once you have all of these little platelets interconnected, they're going to undergo what is called platelet degranulation, meaning they're going to undergo exocytosis of all of their granules and all of their other clotting factors. So they're going to release all of the chemicals that they have stored at this one small site. Now, some of the chemicals are things like serotonin, which, as I mentioned in the vascular phase, can actually intensify vascular spasm. Your platelets will release ADP. Now, ADP was introduced in A&P1 as what was needed in order to form ATP. And then when you use ATP, it breaks apart into ADP and phosphate. So ADP does have a function with energy storage and energy release. However, when it is released by a platelet, it's actually considered a clotting factor. This ADP has the ability at this little wound site to actually attract more platelets and trigger those platelets to undergo degranulation as well. Another chemical released by your platelets is thromboxane A2. This particular chemical also promotes platelet aggregation, so the accumulation of platelets in your plug. It encourages platelet degranulation and encourages the vasoconstriction, so it does also intensify that vascular spasm. Now, this platelet plug formation, these three steps, they actually run by positive feedback. So think about it. You have your initial group of platelets attach. They make their connections with each other and the vessel wall, forming this really small platelet plug. Then all of these platelets undergo degranulation to release their granules, clotting factors, and so forth, including that ADP. Now the ADP in that area causes more platelets to adhere. Those new platelets establish cytoplasmic connections with the other platelets in the plug and the vessel wall. This makes your platelet plug a little bit bigger. And those new platelets undergo degranulation, releasing their chemicals into the environment, including ADP. That ADP then encourages more platelets to adhere, making your platelet plug a little bit bigger. They develop their cytoplasmic connections, they undergo degranulation, more ADP gets released. Can you see the positive feedback beginning? And every time you go through those three steps, The platelet plug just continues to grow and be more effective at reducing or stopping minor bleeding at that site. However, if you think about positive feedback from A&P1, positive feedback does not stop on its own. It will continue to run and to run and to run unless it's interrupted by an outside force, which is why positive feedback can be life-threatening because it will continue to push you further and further away from homeostasis. So think about this situation. You have started a positive feedback mechanism. This platelet plug is continuing to grow and grow and grow. Do you want it to grow into healthy areas of that blood vessel? Do you want it to become so large that it now is blocking blood flow into other tissues? And the answer is no. So you have to have some way of controlling this positive feedback mechanism so that you form the platelet plug where you need it and at the size that you need it. Now, prostacyclin is actually one of the chemicals that does help to limit it because, as I mentioned at the beginning of this slide, normally your blood vessel wall is nice and smooth and covered in prostacyclin, which is a platelet repellent. The only place where the lining of the vessel is sticky and rough is at the wound. All of the healthy areas that you can see in that figure on the screen, all of that blood vessel extending to the left, where there's no platelets adhering, that surface is smooth and covered in prostacyclin. So that plug cannot continue to grow because it can't get a foothold. It can't attach. There are also in your plasma a lot of inhibitory chemicals produced by your white blood cells that do also interact with different components of this process, helping to limit the positive feedback. Especially the plasma enzymes that break down ADP. If you can break down the ADP, you can limit your platelet adhesion. So by taking ADP out of the mix, except for just at that wound site, definitely make sure that that plug cannot grow inappropriately. Also, when we complete our platelet plug, we shift into our coagulation phase, where we actually transform our plug into a clot. And the clot tends to isolate the plug from the surrounding environment. And when you isolate that plug, you isolate it from all the chemicals needed to continue plug formation. Alright, so now we have our platelet plug. We're ready to transition into our third and final phase, our coagulation phase. Now this phase takes a little bit longer to occur, so it doesn't start until about 30 seconds or more after damage was initiated. The goal of your coagulation phase is to take fibrinogen, that nice plasma protein from your liver, soluble in water, and change it into insoluble fibrin threads that will help to form your clot. And this fibrin is going to help your formed elements adhere. So when you hear the word clot, think about a giant mesh of fibrin threads with formed elements trapped within it. This clot, the fibrin mesh within it, is so incredibly tightly woven that the only thing that can pass through a clot is something called serum. Serum is plasma minus your clotting factors. So the only thing that can actually squeeze through a nice dense clot is the watery portion of your blood. The platelets, red blood cells, and white blood cells will not be able to pass through that mesh. Now, the coagulation phase is very involved. There are over 30 different chemical reactions that are going to occur, numerous clotting factors. The good thing is that for our A and P, we don't require that level of detail. We're going to give you some, but definitely nowhere close to even half. So what I thought I would do is give you a little clotting summary first, so you can see where we're going, and then we'll talk about the mechanisms with a little more detail. Now, when we begin the coagulation phase, we begin with two mechanisms. They can run one or the other, or they can both run at the same time. We have an extrinsic mechanism and an intrinsic mechanism. Now, each mechanism has its own series of chemical reactions. However, the end of both mechanisms is the same. Both mechanisms are going to cause the formation of something called factor 10. Factor 10 then causes prothrombin activator formation. Now, prothrombin activator is also called prothrombinase. Prothrombin activator, aka prothrombinase, is a very active enzyme. So the goal of your extrinsic and intrinsic mechanisms is to form factor 10 and then have factor 10 cause the formation of prothrombin activator. Once you have prothrombin activator, you now enter the third and final component of coagulation, and that is the common pathway. Prothrombin activator is going to change prothrombin, which is an inactive enzyme always present in your plasma, and it's going to change that inactive enzyme into a highly active enzyme called thrombin. Thrombin then can catalyze the reaction that changes fibrinogen, your soluble plasma protein, into fibrin, the insoluble threads that form your clot. So now let's take a little closer look at the extrinsic mechanism, the intrinsic mechanism, and the common pathway. Now, the extrinsic mechanism occurs rapidly. It occurs within seconds after damage. And in the extrinsic mechanism, tissue thromboplastin, which is also referred to as factor 3, leaks into your blood from cells outside the blood. So what that means is that damaged tissue, so either damaged tissue in the blood vessel wall, damaged tissue outside the blood vessel, so damage to your epidermis or to adipose tissue, some other tissue, the damage here is going to cause them to make tissue thromboplastin. The tissue thromboplastin then diffuses into your blood. This initiates a series of chemical reactions that does involve calcium. Calcium is considered a clotting factor. And at the end of this series of chemical reactions, factor 10 is formed, and then factor 10 allows for the formation of prothrombin activator. Now, the intrinsic mechanism actually occurs more slowly and actually takes several minutes to occur. Now, this mechanism is actually initiated by the exposure of platelets to the collagen fibers in the basement membrane at the damaged site. Remember, back when we talked about vascular spasm, I mentioned that the endothelial cells contract and expose the basement membrane to your blood, especially the collagen fibers. And those little collagen fibers can actually cause platelets that are passing by to release their contents. It can cause platelet degranulation. Now, during this degranulation, we release a clotting factor called factor 12. Factor 12 initiates a series of chemical reactions that are also assisted by other chemicals from platelets like PF3 and calcium. And at the end of this series of chemical reactions, we have the formation of factor 10, and factor 10 then causing the formation of prothrombin activator. Now, once we have our prothrombin activator, as I mentioned on the previous slide, we initiate our common pathway. Prothrombin activator converts the inactive thrombin in your plasma to change into active thrombin, okay, a nice active enzyme. The active thrombin enzyme with calcium present converts fibrinogen into fibrin threads that are then going to form a nice dense mesh that help to trap your formed elements, form a nice sturdy clot that effectively stops your bleeding. So what happens to a blood clot once it forms? Well, after the clot forms, it's going to undergo what is called clot retraction. Clot retraction is a consolidation or a tightening of the fibrin clot due to the actions of the platelets. So remember, your platelets formed this interconnected mesh where they were connected to each other and to the vessel walls. And after the clot is formed, the platelets all contract and make your clot smaller, and they pull the walls of the broken vessel closer together. So it's not just making your clot smaller and tighter, it's making the actual wound itself, the opening in the vessel wall, smaller as well. And there are benefits to this, because a smaller clot in a smaller wound is going to be a lot harder to dislodge, so it makes the site more stable and it makes rebleeding, opening up the wound again, harder to do. But by bringing the walls of the vessel closer together, you actually can improve repair. It's going to be easier to reconnect the broken edges of the vessel if the edges are actually right next to each other. You'll also start to have platelet-derived growth factor in the area. Now, as the name implies, this growth factor is made by your platelets, but also by your endothelial cells. And the purpose of this growth factor is to stimulate your fibroblasts and smooth muscle cells to undergo cell division. So we're going to start increasing cell numbers. The increase in the number of smooth muscle cells is going to help repair the muscular layer of the blood vessel wall across that break. The fibroblasts that you are going to be forming can actually invade your clot. They can produce a real fibrous connective tissue within the clot, which is going to help to strengthen it and also help to seal the vessel. Now, you don't keep your clot forever. The clot is going to have to be destroyed once repair is completed. To do this, we're going to undergo a process called fibrinolysis. Lysis means destruction. So we're going to start destroying our fibrin. And in order to do fibrinolysis, we're going to have to take an inactive enzyme in your plasma, which is called plasminogen, and convert this inactive enzyme into a highly active enzyme called plasmin. Plasmin has the ability to dissolve fibrin, meaning it can dissolve away your clot. So after your vessel wall has been repaired, plasmin basically comes in and starts eating away at the edges of the clot until the entire clot is basically dissolved and removed. Now, the formation of plasmin is due to the presence of various chemicals. The factor 12, which is released at the beginning of your intrinsic mechanism, factor 12 causes the formation of a chemical called kallikrein. Kallikrein causes plasminogen to be converted into plasmin. Thrombin, the enzyme that helped us to form our clot, also at the exact same time causes the formation of plasmin. And then there is tissue plasminogen activator, or TPA. Now, TPA is produced normally by tissues in the body when a clot is needed to be removed. However, it can also be given medicinally, and we'll be talking about that in a little bit. Now, what's interesting is that plasmin indirectly promotes kallikrein production. So all you have to do is have factor 12 released with the intrinsic mechanism, form a small amount of kallikrein, which then causes you to form plasmin. The plasmin then causes you to make more kallikrein, which then causes you to make more plasmin, which then causes you to make more kallikrein. Can you see another positive feedback mechanism occurring? Guaranteeing that you're going to have plenty of plasmin available to help destroy your clot after repair is completed. Now, there is a dietary component to all of this. You need to have vitamin K in order to undergo your coagulation phase. Vitamin K is used by your body to produce 4 clotting factors. Now, roughly 1/2 of your vitamin K is produced in the large intestine by the bacteria normally residing there. The other half of your vitamin K is ingested with your diet. Now, vitamin K is a lipid-soluble or fat-soluble vitamin. Vitamin K deficiency leads to an inability for you to clot your blood. Because remember, vitamin K is needed to produce 4 clotting factors. And when you start thinking about clotting, the coagulation phase, as a series of chemical reactions, it's like trying to climb a ladder. That every rung in the ladder has to be there for you to get from the bottom to the top of the ladder. If vitamin K is needed to produce 4 clotting factors, if you don't have vitamin K, you are missing those 4 clotting factors. Those clotting factors are missing rungs on your ladder, and it's going to prevent you from actually completing the coagulation phase. Now, vitamin K deficiencies can very easily occur if you have any problem in the small intestine absorbing lipids. You will have a dietary vitamin K deficiency. Vitamin K is lipid soluble. It gets absorbed the same way that you would absorb steroids or fatty acids. So if you can't absorb lipids, you can't absorb vitamin K. If you just have a dietary deficiency, meaning you don't eat the nutrient sources that have vitamin K. If you are on antibiotics that killed your intestinal bacteria, because remember, half of your daily vitamin K is produced by the bacteria housed in your large intestine. You kill those bacteria, you now have a vitamin K deficiency, which is why they often will recommend with antibiotic treatments that you not just take probiotics, but you also supplement with vitamin K to make sure that you've got some extra K coming in with your diet to try to counteract any intestinal deficiency. Now, the coagulation phase has to be very tightly regulated because just like the platelet phase, it runs by positive feedback. Thrombin, along with factor V, increases prothrombin activator production. So think about it. You start your common pathway. You have prothrombin activator converting prothrombin to thrombin. And the thrombin you just made causes you to make more prothrombin activator. More prothrombin activator just makes more thrombin. And it goes back and forth in this positive feedback loop. So you run a risk of your clot growing exponentially into healthy areas. So how does your body prevent inappropriate clotting from occurring? Well, don't forget what we talked about with fibrinolysis. Thrombin activates plasmin. Plasmin is the enzyme that can actually dissolve clots. So if your clot is attempting to grow into healthy areas, you can actually have plasmin present to help eat the inappropriate growth of that clot. And don't forget, plasmin in turn promotes the formation of kallikrein, and then kallikrein in turn leads to the promotion of more plasmin, promoting that positive feedback mechanism. So forming a clot automatically causes you to produce high levels of an enzyme, plasmin, that will limit its growth so that it grows where it needs to be, but not where it's inappropriate. Now, your body is not perfect. Things happen. So you don't want to have just one way of controlling positive feedback, not with something so important. So you have multiple things that you can actually use to help control coagulation. For example, platelet repulsion. Remember, platelets do not adhere to the smooth prostacyclin-coated walls of undamaged vessels. So if your platelets can't attach to the wall of the vessel, you can't form a platelet plug, and if you can't form a platelet plug, you can't initiate that coagulation phase. The fibrin that you are forming will actually absorb thrombin into the clot. So the enzyme you need in order to form fibrin is being sucked out of your plasma by the fibrin itself. And if the enzyme is hidden away inside the clot, there's no way for it to stimulate more fibrin formation. Prostacyclin, again, I mentioned, is that platelet repellent coating that's on the inside of your blood vessel. It's made by your endothelial cells and your white blood cells. But I wanted to mention it does more than inhibit platelet aggregation. It also opposes the actions of thrombin, ADP, and thromboxane A2. So it has the ability to limit the ability to do the platelet phase and your coagulation phase. You then have anticoagulants. Anticoagulants are substances that can delay, suppress, or prevent blood clotting. Some of them are natural, some of them are not. There are two natural ones that I want to mention. There is antithrombin, which is made by your liver. Antithrombin can actually deactivate thrombin before it can even act on fibrinogen. So it can stop your coagulation phase right before the clot even forms. You then have heparin. Heparin is made by basophils, which are a type of white blood cell, and mast cells. Now, mast cells are inside your body tissues. They are relatives of the basophil. Heparin, when it's produced, interferes with prothrombin activator formation. It can block thrombin action, and it also promotes the function of antithrombin. So it makes antithrombin even more potent at deactivating thrombin before it even can activate your fibrinogen. With regards to medications and other chemicals, coumarin and coumadin are very closely related. Coumarin is a sweet-smelling extract from tonka beans and sweet clover. It does take up to about 2 days to act when given medicinally to a patient. Its mechanism of action is to act as a vitamin K antagonist. So what coumarin does is it prevents vitamin K from performing its normal functions in the human body. So it puts the body in vitamin K deficiency, meaning you lose your 4 clotting factors and you can no longer undergo your coagulation phase. Coumadin, which is referred to as warfarin from the pharmaceutical industry, also acts as a vitamin K antagonist. This is the one that is given more readily as a medication. Aspirin is an anticoagulant. Aspirin actually suppresses thromboxane A2 formation. And by blocking thromboxane A2, aspirin can inhibit platelet aggregation and inhibit vasoconstriction. So it decreases vascular spasm and prevents the formation of your platelet plug. So aspirin is a really good preventative as long as you are working with a physician and they are monitoring you. But taking an aspirin every day, people will say it thins your blood, but what it's doing is it's keeping your vessels more dilated, which is going to prevent that nice narrow opening that promotes platelet aggregation, and it just prevents aggregation altogether. So great at preventing clots from forming. Hirudin actually comes from leeches. They have discovered a lot of useful anticoagulants in animals that feed on blood because they have to keep blood flowing as they are sucking it out of their prey. And they're starting to try to isolate these chemicals to see how we can use them for us. Hirudin from leeches actually inhibits thrombin formation. So it prevents your coagulation phase from running. Hementin also comes from a leech, and hementin actually has the ability to dissolve clots. And they have had some real promising tests with hementin in cardiac patients with dissolving clots in blocked coronary arteries. So there are lots of really promising applications for hementin coming down the pike. Arvin is actually from the Malayan viper. So this is in its venom. And what they have found is Arvin rapidly breaks down fibrinogen. So it actually breaks down that plasma protein. And if you don't have fibrinogen, you can't form fibrin and you can't form the clot. So there is some potential here. Streptokinase is actually produced by streptococci. So it's produced by that strain of bacteria. And streptokinase digests proteins. So it's just a generic, it breaks down protein, especially fibrin. So it does have some clot digesting applications. You can even be given TPA, tissue plasminogen activator. It is made by the human body, but you can receive it in an injection, much higher concentration, to activate higher levels of plasmin in the body to help dissolve inappropriate clots.